full of large boulders and blocks.
So then just turn it on. There. And now you should be able to go back to live streaming. And then that should start coming in.
But
Hey,
the
Steve. Oh, go ahead.
no, I was noting that some of these large boulders are quite large. There's still lasers on the screen if you can see at home, 10 centimeter lasers that are kind of dead center right now. Their color is green, so it might blend in some of the warmer parts of the frame, but you should be able to see it.
Just real quick.
So Steve, we should share the conversation that you and I were having earlier about trying to think about what the age of the seamount might be. We were looking at some pretty cool movements of the seafloor over millions of years, right? And what did we get to? Around 80? You'll have to.
Yeah, well, it's interesting. So most of us, if you're interested in the marine environment, you've probably heard about plate tectonics, or if you're interested in geology, you might have heard about plate tectonics and how plates move across the Earth like, kind of floating islands on the sea of molten material below the crust. So, in a lot of these seamounts in the Pacific are formed by hotspots, seamounts and islands for that matter. The underlying geology are formed by hotspots, volcanoes that basically eject lava through the crust, forming, these large mountains. And it's possible to date some of the rock. Some of our geologist colleagues on shore are interested in some of the material we have here to both try and assess what is the seafloor made of, and also how old the rock might be. And generally you find a lot of the older seamounts in the Pacific are in the northwestern part of the Pacific Ocean, and they get younger the further south and east you move. In this area, there's some very sparse evidence that the ages of these seamounts might be in the 80 to 100 million year old range as we get down towards the Line Islands. So if you were to follow this seamount to the southeast, it kind of blends into the chain called the Line Islands. But it's very complex seamount geology and plate tectonics in the Pacific, because you don't know, which seamounts might belong to which hotspots. The hotspots are generally fixed, have eruptions and volcanism occurring on a seamount from several different hotspots. So one of the goals is to collect some rocks here as we're going up. We started with one at the seafloor where we set down. But yeah, so we'll be able to, hopefully, if we have some good basalt rock on the rock we just collected from our first rock collection, we should be able to send that off to our colleagues to get aged and give a better idea of how old this seamount might have been.
So we just got a question from Kiribati behind the scenes here. I don't know if you can hear me, but they said, is this the seafloor or a seamount? So it's important to clarify that this is the seafloor on a seamount, which gets even more confusing. And by seafloor, it's not the floor that you and I are always thinking about. It's actually on an angled up. It's hard to tell from this point of view. But I think it's really cool to sort of envision yourself on an upward slope on the side of a mountain, rising up from the bottom of the sea.
Wait, this is the seafloor?
Yeah, the seafloor of the seamount.
Okay, either one.
So there's a couple of terms we should clarify, namely that the surrounding there's a deep abyssal plain in the Pacific. The flat, some might call featureless, but it's actually really complex environment. Very flat terrain.
Oh.
Yeah, there you go.
That's the first shrimp we've seen.
Third by my count.
Third?
Yeah.
Oh.
Yeah.
We don't
But
have
just
Third.
maybe Tim might be able to chime in from the library. I could probably make a guess, but
Yeah, he says it's a nematocarcinid trim.
They typically have these longer legs. I'm not sure. Get some more info from him, Tim, later.
So I was expecting to see a lot of like corals and stuff down there, but it seems like you barely see stuff. Why is that? Like,
It's a combination of factor. Crusty stuff. Oh, and the sponge there. Is that what you're pointing at?
the white thing?
Yeah, it looks like it could be a sponge.
Oh, okay.
I think it's a sponge.
Yeah, it's like a sponge. Spongy.
Do you know what kind of sponge?
Sponge in a really nice spot, actually. Looks really secure.
Except
And it's like, yeah.
she can't hear you every time you speak, so
No.
you gotta be either louder or closer. Your voice is so low.
Okay. Cool. I don't know because I was like trying to speak louder, but probably my voice is so.
I'm Miss Rocio.
So what we were talking about earlier, did we get the image on that?
Yeah, I got it.
Ready to go. Whenever you're- we were talking about earlier why we don't see more stuff, like why we
Yeah.
don't see more corals, why we might not see more sponges, and generally the rule is that you see more stuff the shallower you are, and the deeper you go, the less larger organisms you typically see. There's life all around us on the seafloor here, in the sediment, in the crevices and cracks underneath rocks. There's life all over, but you don't see the large corals that you might, because in reality the animals that live here that are larger generally live on particulate material, so particles that drift down from the surface of the ocean, marine snow, basically we call that. And as the marine snow, it's relatively fresh, it's full of fresh, plankton up in the photic zone, and as it starts to sink down, there's a couple things that happen that microbes will respire and consume the organic material of the marine snow. Therefore, they take that carbon, that energy, that they take it in, they respire CO2. CO2 goes back out into the water, and the carbon quality of the food is reduced the deeper and deeper and deeper it goes, because the microbes have more and more time to eat the food on the marine snow. And so by the time it comes down to 2,000, 2,500, keep on going down, it becomes lower and lower and lower quality over time. It's a nice
I bet you there's
big
gorgeous
sponge.
stuff at the top. Look
Big
at all these
sponge
things on the
debris,
bottom.
yeah. So we did mention we started on a slope, and the slope, generally things go downhill here. Gravity still works underwater, so things will fall down slope, and you'll get an accumulation of debris. You might see sometimes where there's a flat ledge, there's more debris than on a steeper slope.
See that little override to the left in the crevice?
Yep, yeah, right over there. It's gonna be a tough grab there, but try.
No, so if you're not losing one of them, so first you're streaming to the wrist, which means that you have to come on street.
That's a tough shot. It's partly blocked, so unless there's a significant interest, I think we'll pass on that one.
Tim wants to be able to talk to you guys in here from the library.
So, yeah, so
I can turn the outreach speaker on, and then he's talking over YouTube and talking to you guys.
This is great.
Okay, so what, answer him, just talk back on that microphone because he's hearing it. There might be a tiny little delay. It won't be instant, but basically I'm going to turn the speaker on up here so that you hear him if he talks to you.
that's
Okay?
great.
Ethan, I told Tim to come on in.
Yeah, you can do that.
I think it's just. The only thing it might have issue with is that one. Hey Tim, could you try saying something? Hi, everybody.
Test 1, 2, 3.
All right. I got it
Great.
too.
Perfect, thank
That's
you.
awesome.
So that you can have that conversation there, okay?
Spectacular.
What is
Thanks,
he on?
Debs.
What is he on?
Outreach. So he's on outreach, and that will be live. So those conversations.
So as we move shallower, I expect we will find more things.
Okay.
Yeah, I think we should just leave it away. You promised me a little.
That being
crazy
said,
market,
you know,
so.
yeah. Should put a disclaimer on all these dives. No guarantee of seeing things.
Hey, I don't know. You said that with really strong.
I still believe it. Yeah.
I do too.
Oh yeah, you don't place a lot.
Yeah. Small,
Oh yeah, a little pouring.
small isopod there.
Yeah.
Mhm.
Menopsid. Yeah, but it looks a little bit more cohesive here in terms of the seafloor. You know, not so much smaller broken bits and gravel.
Yeah, if we had to collect a rock here, it would be really hard.
It would.
Oh, look at there. Yep. Okay. Buttonology. No Freud. I'll get that one.
I'm gonna keep my radio, okay?
Oh, you don't have to put a sample number for observations. You can just put. Yeah, get rid of that. Just, you don't have to put anything. You just say, you know, hit the fish button, hit submit. That's okay. Yeah. All right. Fish is gone.
Oh.
Yeah. Only things that go onto the vehicle get sample ID numbers.
Okay.
Okay. Call me if you guys.
Oh, there's a coral to the right.
Yep. It looks like another Candidella, Prim, Noid, Octa Coral. We got some really great imagery of one. Actually, another couple maybe.
Which one you want? You want to go look at one? Or?
Associates, right?
Oh, the one on the left looks a little different.
Okay.
Can we look at that one? Yeah. I'll be able to tell you in a second
Okay.
if it's worth. No, it's the same.
Roger.
Yeah, that's okay. You keep going. Yeah, so the habitat here is a little bit more cohesive, but you know there's some areas where it looks pretty well cemented. The rocks look pretty well cemented to the seafloor. For folks on shore that are writing in about the environment, it's presumed that these seamounts are, you know, very much extinct, and that, you know, there's no active venting going on here. We don't have, you know, we're not really near an edge of a tectonic plate where you get venting going on,
Look at all these.
or near some sort of fault boundary.
Oh. There's some interesting things right here.
Yeah, there's a few things here. They look like sponges of some type. I think that.
Did you see the star over here?
Star? Oh yeah, yeah. There's a few brittle stars there. You can see the.
Every time you talk to Kanji Tech, it tells me you need to speak louder.
Okay.
You wanna? Can we?
I have no idea.
It's better.
Okay. Yeah. So we were just looking at some brittle stars here on the rock face. It looks like there's some nice. Looks like some nice, you know, bumpy, crusty accumulation on the vertical surface of these rocks. It looks like a lot of these sponges too are, you know, they're not looking the healthy. You notice that sponge we looked at earlier that had the nice, very
Yeah.
a lot of sediment over time. But yeah, this is what the rock looks like without the sediment drape. You can kind of see, you know, this is more a vertical surface. Not a lot of sediments accumulating here.
Oh, it looks like another one. Wait, just
Oh,
a stick?
Yeah, that one might not be a live critter anymore.
It's dying.
Can we look at the stick on the? Yeah, kind of right there. I'm just gonna zoom in quick. Zoom out.
What's in the bottom right? You see it next to it?
Like the red thing?
Yeah. That's all right. Don't worry.
Maybe it's a shrimp.
Yeah, I see it.
Couple of shrimps.
I think it's right there. Oh, no.
Yeah, but
You wanna look at both of them, or?
just the one that's right near that might be. It's a combination of things. There's and then a lot of what we're seeing immediately on top of it is crust. So this is the iron manganese precipitation onto the rock surface. So that in some cases the crust can be quite thick, several centimeters, and the accumulation of that crust is on the order of millimeters per millions of years.
Yeah.
So very slow process. And then the white that you see quickly. There you go. So yeah, this looks that's a holothurian, a sea cucumber. Yeah, right where the lasers are right now. You can kind of see the intestines are wound up, and the mouth parts are facing up and towards us.
Do I need to press something about it?
No, I got it. I'm logging.
Okay, cool.
You can just enjoy. I'll log.
Okay.
All right, looks good. Continue.
Takate Teke says she still can't hear you. You need to be louder. She
Yeah.
can hear Steve, but not
For
you.
reals?
You could just say hi to her so she'll feel better.
Hi, Tekatiteke. It's so hard to pronounce your name sometimes. I'm pretty sure if she can't hear me, it probably the internet.
No, she can hear Steve.
Really?
Yes, your voice.
Okay.
You have this beautiful, deep, low voice, which I really love, but it's hard for people to.
The goal of the first waypoint was to get on top of this ridge, right?
Yeah.
So we're gonna do a slight direction change when we reach our first waypoint. Proceed. There, yeah. But we've come up about 100 meters actually since we. Or no, wait. Let me see. Where do we go on bottom here? 2210. No, almost 100 meters we've come up. 80 meters so far. And it seems like the seafloor is rising pretty fast, so we should, as we approach our first waypoint and beyond, we'll be coming up a little bit more in depth. So on the is that bathymetry live right now? That's yeah. Yeah. So on the bottom center of your screen, you can see the seamount that we mapped over the course of. Did a few passes over, and we're on the ridge that's closest to the bottom of the screen right now. And we're headed up towards the top of the Guyot summit, a small ridge that's in this feature. There you go. You can see our track line now. So by the end of the dive, we're hoping to be in the. Let me see.
Talking to me? No, it does on that one has
Okay.
been printed. Sorry, the new one is here on the printer for you. Sorry.
So you're just like looking for specific things, not all of the stuff that you've been seeing in the.
Well, we're trying to look at and get it, characterize the environment right now. So we're looking at everything.
Yeah.
We're trying to look at everything we can look at. There's some things that are just too small that we won't be able to see very well, and we're just trying to look at larger things right now. So megafauna is the category we use. Just basically things that are larger than a few centimeters. Is plenty large enough for us to rocks. There's probably worms and all sorts of other small crustaceans.
Yeah.
The waypoint
Yeah,
should be at 2019.
perfect. For number 2. Yeah.
What's that thing?
Looks like some debris here. I think, this actually looks more like
Looks like sticks.
coral debris. So yeah, we were seeing a lot of sponge debris. The first leg of this dive between our on-bottom target and waypoint 1 here. But we're looking more now at what looked like possible coral debris, these things that look like sticks. But we'll see when we get, thank you. We'll see when we get, as we continue to move up slope, hopefully we're going to get a handle on what's making some of this stuff. There we go. Yeah, this looks like coral debris. This is kind of the fun part. It's a bit like a teaser of what you might find later in the dive.
Mm-hmm.
I'm not logging dead skeletons. Should I?
It's tough to tell what it is right now.
What? I see dead things. Captain Al has the answer. He says, right, I see dead things. I'm waiting.
Some nice, look at this. These rocks. There's a slight overhang here.
Oh, this is blue, right?
The what?
The sand sponge. The sponge.
Yeah, that's what I was mentioning. The sponges kind of look, if you look at this sponge right here, that's kind of the center left versus the far left.
Mm-hmm.
That sponge looks more, transparent, translucent,
Yeah.
and that sponge is more opaque. It sometimes can tell you something about if the sponge is live, dead, how it's feeling.
So there's like a different kind of sponges as well.
Yeah, but there are other kinds of sponges that are entirely opaque, and they can be very much alive.
Mm.
It's actually, while I'm over here, I'm going to look at just this piece, because this is evidence of some coral we haven't seen before. But it's a dead piece of what looks like, this looks like it's an Octa Coral, but it looks like it could be a type of hemicorallium. And I'm hoping to see some more as we...
This crack right here, see that little pink thing in there? That crack? Bottom left screen.
Yeah, I got it. Oh yeah, it's a siphonophore.
Oh.
Yeah, so benthic siphonophore. Yeah, these critters are suspended by these fine anchors. Portuguese man of war, jellyfish. They're slightly different from the Medusa jellyfish you might see, but those types are from the surface ocean. These you see quite a bit. Awesome colors and shapes that these things can occur in. In fact, the longest organism in the Falkor and SuBastian off of Australia. That was a siphonophore. This is a similar, same type of organism, different species. Yeah. And these are quite common down here.
Do you want me to press something about it?
I think that's fine. I'm sure we'll see some more. But that one was really nice because it was well anchored in that crevice. Basically they can anchor themselves, and they're feeding on, again, particulate from the water column. There's actually quite a bit more marine snow here than when we started. I don't know if you noticed that, Randy. The looks like fuzzier, cloudier in the water, which could be an indicator of higher quality food or more food. Yeah. So that stick we were looking at earlier makes sense now that it was probably a part of a precious coral skeleton, and I'm hoping, since that one looked about the same, that we might see some more as we move up. In fact, typically they like these kind of ledges and tops of these ridges because they has good water flow over them. I wouldn't expect to see them growing in these cracks and crevices of the rock, but as we move up and on top of where there's fresher flow, where they have an easier time feeding, we might see more of them, or more lively ones.
This expedition, like, you guys may be probably like talking about some species that they are gone already, and then once a while when you do the research, you found them, like some of them still alive at some point.
I don't know of any off the top of my head. A lot of the things that we find down here are just completely new.
Oh,
Like,
okay.
we didn't know that they existed before this because we either don't have the technology to environment that's hard to hard to see, or hard to collect. A lot of what we know about deep water corals and sponges earlier on in history was from what was brought up in fishing nets and things like that. Obviously there's no fishing going on here, at 2,000 meters. But it's hard to see, if something's in a crack or crevice, how that could be sampled without technology such as this. Yeah, thanks for everyone writing in about the dandelion siphonophore. Common name for that cool little siphonophore we were looking at earlier. So some pretty massive, what I think are sponge stalks, sponge skeletons.
I can't wait to see what's above. I bet we're gonna have maybe a great forest.
There's no telling how old these could be too. They could be here for decades,
For a very long time.
centuries. Yeah.
Interesting. So you just check it out from the bottom to the top. That's how it goes.
Yeah, we're moving up slope, starting deeper and moving more shallow. It helps us get a better picture of the seafloor than if we were going, say, down the slope. And see how it might be dangerous if you're, going down slope in an environment like this. You want to keep this terrain in your front view. Yeah, but it's slow going. Especially when you have terrain like this, you don't want to move too quickly, or you can put yourself in a very precarious situation.
It's funny because I was about to say speed up.
Yeah. We can speed up. So what are we doing now? 0.3?
0.1.
Oh yeah, we can speed up if you're comfortable with that.
Yeah, I'm just coming up this wall basically. And
Yeah.
just to be very careful. We flew into 2 caves the last time we were
Yeah. No, that's definitely
So we just want to make sure.
definitely a possibility in any of these seamounts.
Can you? sorry.
Like I can back out and kind of boot you up a little bit.
No, I think we're moving at a good pace here. You know, while we move up across the steep stuff and
Sponges are about to start getting more.
yeah.
We got 3 of them in a row right here. So.
That's a sponge too.
Yeah, that looks like a sponge.
Can you mark things for a little bit? I'll be right back.
It's like,
I'll get the sponge
yeah,
out.
you got a wall.
Yeah, so definitely more of these large glass sponges and different sponges than we were observing deeper as well. A lot of the corals that we were observing when we first touched down are, you know, not seeing too many of them. So far we've seen, just to recap, we saw a few corals. We saw a couple different types of Prim Noid Octa Corals. We saw a couple different types of black corals. So far we've seen a number of these sponges, glass sponges, several different species. Hit sponge?
Sponge?
Yeah, sponge. And just hit, yeah. That's fine. We can always fill it in later.
Okay.
Okay, so about 50 more meters to get there. It looks like it's about 50
This
more
is a
meters.
sponge too.
Yeah, those are all sponges. Those more
glass.
opaque ones, yeah, they look probably, you know, like this one here. This one looks more lively,
Hmm.
but the ones that are, you know, more opaque, or, you know, they look like they are, you know, brown, or they're filled with sediment, probably dead and, you know, hanging onto the wall by their skeleton.
Hey, the current
So
wave
we are
out there, I'm out of your stop.
looking at some of the navigation, and we have about 50 more meters until we're at the top of this ridge, and then we're going to continue up along this ridge. We're on the steeper slope of it right now on the side, and we're hoping to we might see some different organisms up on the top of the ridge, since the morphology of the seamount is slightly different up there. Here it's more steep. There probably be a little bit more gentle slope.
That's why I saw that. You got to go around.
It's really one of the things I always try to keep my eyes out for in these, you know, deeper dives are small, you know, small things that you could easily miss. You know, small translucent or transparent, you know, small.
Even if it's just a dot like this one.
Yeah, you know, sometimes if you see something, you can see something waving in the current. That might be a coral stalk, or
Okay.
you know, a lot. You know, sometimes you might find things that are colorful down here, but typically the deeper you go, the less color there is for corals and sponges. It's all kind of, you know, pretty, you know, pale or translucent. But we did see some pigment in one of the black corals earlier. It looked slightly pinkish, reddish pink, brown.
Oh, there we go.
Yeah, we're getting up there. Seeing more sponges now, adhering to the.
There's a lot of sponges
Yeah.
everywhere.
They seem to like these vertical, steep surfaces. You can see a good selection here of what look like dead sponges versus
Yeah.
live sponges. There was a dive I don't know, it was
Oh,
something swimming up there.
yeah, something swimming around. Probably shrimp.
None of these sponges have any associates, do they?
I
can't see.
haven't seen any.
I've seen some
Yeah.
in some of the other sponges down below.
Yeah.
You
Like
have?
like the wedding sponge that I first saw. There were two
Yeah.
types of that.
Is that
Yeah.
spongivorous? What's going on to the left there? Let's see. Is that sponge destroyed? Which one?
3 o'clock or 6 o'clock
Yep.
or 9 o'clock.
Take a close look.
This one.
Yep. Nope. It's just a
Yeah.
destroyed sponge.
There's a little something living in there.
So
Yeah.
he's exactly 10 centimeters across or so then, right?
Yeah. Yep. Okay. Sure.
Did you want to get a closer look at this?
No, that's fine.
It looks like there might be a coral coming up. Could just be some- Yeah, just straight up.
Oh yeah, there you go. We can stop and take a better zoom on this for a quick second,
Frame it out.
since it's one we haven't seen yet.
Is that my primate on top of that dead sponge? Okay.
Yeah, I think so. There's a few colonies here. Yeah, so
There's like 3 of them I can see. With that same one.
Started to have a lot more.
let's see. Whichever one. Yeah, let's go this one's closer.
Just gonna take 2 seconds.
Sure. Mhm.
Nice.
So what we're looking at here, this one is a Chrysogorgid Octa Coral. Well, yes, that's right now. So this group, this genus was recently revised in a paper that was came out just a few weeks ago. In fact, maybe months now. It used to be called Pleurogorgia.
Hold on real quick with the camera.
Just
Bring it here.
Let them get stable.
Sure.
And then,
You want me to press on it while she was doing something? No.
I think she's got it.
Okay.
So I'm sorry. What kind is what kind of coral is that? Too? Is that what is that?
It's an octocoral. We used to call it Pleurogorgia, but depending on a couple of things here, it was recently revised, and I have to take a closer look to get a better handle on if it could be one genus or another.
I'm just gonna free fly it across. Come straight up.
So night. This is really shaping up here. Look at that.
If it's a
You can
bad
see
spot, there's some more up top.
colors.
Yeah. So what Tim was noting was that when we were here in not here, but when we were in Phoenix Islands in 2017 on our first exploration of the deepwater corals of the protected area, we had noticed that in Pleurogorgia there were really very few or no associates. Associates being ophiuroids, shrimp, crabs. These types of organisms associated with these coral colonies. Gorgids are actually quite abundant on vertical surfaces, much more than in more moderate slopes, which is really interesting. In fact, you can actually have dozens of colonies per square meter. In some cases, we've found of this one species. So they can occur very high density, and if you notice that some of the colonies are quite large and some of them are quite small. So there's probably, Randy, as you were mentioning, a few different recruitment events here for these critters. So you see, these are all the same kinds of coral. 1, 2, 3, 4, 5, six colonies in view right now, but they're all at various sizes, possibly very different life stages.
I don't even know how to mark
Can I
this.
go in?
There's so many sponges.
Sure.
We should ask Brian. if
Should
he
be able to.
knows
I
how.
want to get that one in the upper left. Is that okay? I should be able to reach that run, right? Since it's kind of.
If you, to sample it?
No, just like to look at it,
Oh, yeah,
because
you're good with that.
I think it looks okay.
That's a coral.
Yeah, it's another kind of coral. So this coral belongs to the same family as the spiral coral we were looking at when we first came down on the seafloor, the Iridogorgia. The polyps are typically very small on these, maybe just a millimeter or two at most. I don't see any associates or any kind of evidence of So Tim was remarking that they hadn't seen very many or any on this coral in the past. So I guess we'll keep going here since we have a pretty good idea of what this coral is. All right. You want to look at the sponges here in more detail?
Yeah, let's take a look.
Okay.
I was just asking Brian if we wanted a sponge too. I don't know. Oh, beautiful.
Let me darken that up a bit.
Sure.
Do we know what
Yeah.
kind of sponge this is? All like demo sponge? You put
Oh, this
your-
is glass sponge. Yeah. Hexagonal
Oh,
sponge.
yeah. I see it now. Okay.
I put I think we can probably get to a family or genus level on this with some of the guides we have on board later on. All right, so get a approximate size. They're in the sponge.
In the sponge.
Yeah, there you go. So that
It's about 10 centimeters in diameter across the
yeah, the main osculum there is about 10 centimeters across. All right, moving on. But it's interesting to note that you find these two organisms, these corals and these sponges, primarily on the steeper, near vertical or vertical surfaces. Although there's a couple of other sponges I noticed, these stalked, bell-shaped sponges as well.
Is that a coral?
That over there on the
Yeah.
right, that's a sponge. That's another type of glass sponge.
Oh, okay.
Yeah. But this one is stalked, so it looks like a coral, right? Because
You review about the corals, the type of the corals.
you can see how it has the main body of the sponge is on the end of this long stalk.
Mm-hmm.
It's the same group glass sponges that these
that one.
other sponges belong to. Yeah. All right, ready.
Beautiful.
So this is probably some of the most impressive high density biology we've come across on this dive so far. Owing, I think, in part to the fact that the seafloor here is a little bit more stable. When we first sat down, we were in a kind of a slope with a lot of rubble and or rocky rubble and debris. Oh, there's a fish right there. Zoom in on the fish.
Oh,
Not seen that fish before myself.
Do you know what kind of fish it is? I have no idea.
it's a prawn.
Well, right now it's an upside down fish.
That's
Tim was.
Hi,
Yeah,
Tim.
hi Tim.
Yeah, just looking at this fish, I don't think I've ever seen it before either.
okay.
Yeah, so it looks like we got some good imagery. We got a good shot of the belly, pectoral fins. Some of the characters we're looking at here for our fish friends, fish colleagues on shore, fish scientists.
Anybody does anyone know what it is?
We were. It looks like someone was typing in slickhead.
Could be a
Sounds
sunhead
right.
fish. Thanks for the help.
Yeah, thanks, internet. But it's definitely definitely playing for the camera here. Awesome. And I'm breaking.
Small sponge at some point for Brian, but I don't think we're in a rush to do it.
Yeah, this is yeah, really nice. I'm wondering if it's around this bit of topography, and that could be fueling a lot of this biomass. That would work. Those Brian, if you're listening, those stocked sponges could work if that fits your needs.
Yeah, he's napping before his shift.
Okay.
So he just wants sort of one representative of what's common,
Mhm.
and Anna I think would like one hexagonal at some point, but I don't think we need to rush.
Okay.
So
Yeah.
if there's an easy one in sight, grab it. If not, don't worry.
I have a strong suspicion this community is going to change once the slope changes.
Yep, I agree.
So if you want something from here, it's best to get sooner than later.
Let's get a very small one because I don't think we want a huge glass sponge.
Yeah, no, that'll be tough to fit in the box.
All right. Oh, and there you have. There's your first change.
No, you gotta.
So
Yeah,
there's your
this
coral.
is
All right,
over
I
on
think
the far side. There's a couple of different corals.
Yeah, I think we should get a small
You want us to
small
park,
sponge before they're gone. Because I agree, it's about to change.
do you want us to park over there with those small coral or sponges over by those corals so you can look at?
Most likely. Yeah.
Yeah.
Let's get a little closer. And I want to build the suspense. You know.
I just like guessing to see if I can get it right. It doesn't have to be.
Oh yeah. See if they're getting in to get close. You might want to.
I mean, you want a representative. It's probably
Anna wants one for micro.
the one there, kind of at center right. Yes, great. Let's see.
Do you have a preference on what quiver it goes in, or?
No preference.
Nope, we're good.
Do you want to? Can I move the camera? It should be sample number 004.
Thank you.
But it's going to be going into quiver number 7.
Yeah.
So we're sampling a sponge here. Environment here. And the more common species of this environment.
We also learned last time that the microbial communities on sponges are really outstanding.
Is it generally all sponges have pretty nice microbe communities?
I don't know.
Or?
We only sampled one hexagonal last time,
Yeah.
and it was one of the most productive microbial communities we'd seen.
Yeah.
So, productive
Well,
in terms of
this will be a
biodiversity.
fortunate collection then.
Let's hope.
So it looks like it might just like perfectly fit in there.
Yeah, I'm just trying not to crush it too much.
Appreciate that.
I'm sorry, and what's it going to go into? What job? Which number?
7.
7.
Thank you.
There you go.
Yeah, very nice collection. Like a glove. So that's in quiver 7.
Hold on. Looks like you might have gotten a bonus coral. Check that out, Steve.
Oh, yeah, right there.
What's going on?
I think it got broken off from that, what I thought was that.
In the quiver.
Yeah,
In that quiver or a new quiver?
new quiver is good.
New quiver. New quiver.
Okay, let me close this one up first.
Sure.
Steve, what is that? Which coral is that?
You know, I didn't. I think it's an Iridogorgia. I didn't quite get a good look at it from
Okay.
you know, we focusing on the sponge first, but we'll get up to that coral after we finish this collection event. Sponge is good in 7, waiting on the lid.
So if you're just tuning in on World Oceans Day, we are here on the inaugural dive of FK210605 Expedition to the Central Pacific at the Phoenix Seamount that we mapped last night and that we are exploring today for the very first time. We are here in what's called an high seas seamount, one that is beyond anybody's borders. And let's see, we have Anamare Tanaba from Kiribati and Steve Askovich from apparently New York these days, and me, I live in Boston. And we have a totally international crew on board, incredible group of ROV pilots, marine technicians, everybody who's helping to make this happen so that we can collect some samples. Sometimes purposefully, like that sponge, and sometimes opportunistically, like this Iridogorgia, which seemed to just hop in our basket. It just really, it wants to be famous for science. It says, sample me, sample me. So
Sample of fortune.
it's kind of perfect actually, because it's, take a look at that. That'll fit perfectly in our respirometer for when we get on deck.
Yeah,
So
no,
great. So we have some really cool experiments that we're going to do on board. Probably at 2 in the morning tonight when everything's up and going. But what are we going to be doing? We are going to be some of these corals and looking at their metabolism and their metabolism in response to just basal metabolism, and then in response to different microbial challenges that we have for them. So we will be doing that work midnight tonight. So if we start to sound a little loopy, it's because we are. That's just part of the fun, right?
Yes.
You tired yet?
No.
Oh, and I see a friend and neighbor of mine is on. Hi, Suanna. So it's pretty fun to see our friends and family watch us in our loopiest, craziest moments. Where are we? 2,080 meters below the surface of the ocean in the middle of the Central Pacific. Why not? Awesome job. It's also pretty cool to have our first dive of the trip on World Oceans Day, and your first dive ever.
Yeah, it is my first time.
Your first deep sea dive.
Yes.
What a great one.
I
You
know.
it's just getting exciting too.
I know. It started off a little slow, but it always does seem to pick up just as you go up.
Yeah. It's always the best rate just before you get off the bottom too. When you have to come up.
Well,
Yeah.
let's try to stay down. This is getting really good.
So right now we're trying to salvage an opportunistic sample, a coral that fell into
A
the
volunteer.
All right. Well,
Good.
I don't want to speak for the coral, but opportunistic sample works for us. That fell into our front porch here, and they're going to try and salvage some of these branch tips. It's not the whole colony, just a couple of branch tips. It looks like, and sample them for a separate experiment we have going on.
Yep.
We're fairly certain that this is an Iridogorgia coral, probably Iridogorgia magnispiralis, but we'll be able to take a look when we go back, zoom out later. Very dexterous movement of the arm here. 005.
FK210605. This is going to be biology.
So by my count, I just see 2 branches.
Yep, I see another one too.
Okay.
It's right
Do
in
you
the middle.
want it, do you want the other one? I can put it in there if you want it.
Yeah, let's do it.
We can go in the
I don't
same
wanna leave it.
in the same quiver.
So I'm just reading the notes that our colleague Dan Wagner wrote to us earlier today, and just trying to put this seamount in context. And it's pretty amazing. He was sort of trying to give us a mountain chain to relate it to. And if you were to put this terrestrially, this would be taller than all but 8 mountains in the Pacific and all but 2 mountains in Europe. It's a pretty tall seamount. And here we are on this very tall seamount in this very tiny area with this, trying to take these robot arms to get this teeny tiny piece of coral. We could probably just leave it on the quivers. It'll probably just come up with us. Trying to grab it from the sense of scale here is just astounding. Probably will just come up with us.
This is like trying to pick up a grain of rice
I know.
2 kilometers away. With I don't know. It's a good metaphor.
Yeah. with I don't know what. With a giant robot claw?
Yeah,
Is that gonna work for you?
That works.
Oh my God! And they got it. Wow. Woo! These guys are amazing.
Very dexterous.
We're gonna call this 004 also.
It's part of the same sample.
Yeah. I love this little comment button where you can fix your mistakes. It's great.
Yeah. All right. Well, that's a good collection. One of them belonging to the sample we just took, and the other a larger sea fan.
Oh, yeah.
Oh, sorry, you want me to move up and right?
Oh yeah, just kind of up and right and out of the frame. There were that one. And that one. Yeah, it looks like the light bar might have dislodged a couple of branches and they fell down to the porch.
Whoa! Look at that. That's great.
Yeah, there we go. So we've got
We got pieces of that.
the subject of our collection. Yeah, we got some pieces of that, an Iridogorgia magnus viralis, and then there's a smaller colony to the lower right.
Hang on, guys. I'm gonna be on the Zoom, so can you guys log? Sorry.
Sure.
This is a great shot of
What type?
Chrysogorgiidae.
This one?
Chryso, yeah, that's right. Yeah, so you can see this this coral sometimes is called Firecracker Coral because it looks like, you know, firework going off. But essentially the coral is made up of all these small polyps, and the skeleton is coiled, and you can see all the polyps arranged on the branches there.
Any coral associate for that?
I don't see any associates. I think there may have been associate, but we might have knocked it off. I saw a small shrimp swimming away, so I would say no associate
Okay.
for now, just to be safe. And we can go look at the other one down to the lower right.
That's another coral.
Yeah, so this is another coral, also a soft coral, much like the one we just saw, but belongs to a different family. So this is going to be a Primnoid Octa Coral.
This one?
Primnoidae, yeah, that's right.
Okay.
And we zoom in, I can kind of show you. Wait to get settled. But what we were saying before about how the currents move around this feature is really indicative, or is indicated in the orientation of the corals. So we were saying earlier, the current is kind of, imagine, the corals here, the current's coming like this, and so it's using the shape of the colony to slow down the water, and hence particle capture. All those polyps are grabbing particles out of the water column and consuming them, both for this colony and the colony above. When you guys are in position.
Any associate for that one? No.
I don't see any associates here either.
Okay.
All good? Alright, we're gonna
Testing 1, 2, 3.
push in a little bit. Do a quick
There's something on
focus. Oh yeah, there might be something there.
I saw something on the ground. Can you help with focus?
Yeah, it's a little sticky. Awesome.
Oh, an A-Plaque down
Yeah, so there's yeah, so what we're looking at here, actually, we're looking at a coral, right? But on the coral, you can see in the upper 2/3 of the frame, kind of in the midline, is a small worm-like creature. It's actually an Aplacophora, a type of mollusk related to clams and snails and things like that. And these are actually notorious coral grazers, coral predators. In fact, I don't know, can I go full in to see if we can get a closer look at what it's actually doing there? Because it actually might be feeding right now. Because you can see the pollen, that's the mouth side. So this is a really interesting observation.
Are you going to get a sample of that?
Yeah, I think we can take a sample. I think that's a fair sample. But the problem is that the grazer's so far down.
I might have to take it in 2 snips.
Yeah, and that's okay. I think that's okay.
Oh yeah, that's
To
fine.
break it up
That's
into
what
2 pieces.
Yes.
Yeah.
But is that,
Can we-
do we want it for the A-Plaque? Is that the goal?
I think it would be a good idea to get the grazer in there as
Okay,
well.
okay,
Yeah. So just the branches from the top part of that colony. If we can get, and we don't have to take the whole portion of that, but at least getting the branch that small creature's on.
Get the creatures.
Yeah,
Okay,
yeah.
I can do that.
Right.
Now that-
So we're going to try and do another sample right now. This will be sample number 006.
Okay, sample in here or this one?
Right there for now. Yeah, sample stored. Yeah. So FK2106-006.
So what we would be,
You can just throw stuff at me, we'll be fine.
So actually, can you back out of that first? Yep. And go to this Ivory channel and put Coral Ivory and just hit submit here. And now you can go back to do what you were doing before.
Falkor 06, right?
That's right. Exactly.
Any type?
Biology?
Biology. Specific location, like storage location.
This is going to be going in quiver 2.
Okay.
Yeah. But hold up. Wait until you see it go into the bucket.
Okay.
So this first piece is the first part of the sample, the same sample,
Why don't you pay me
and
pay me
this piece doesn't have the coral lavore on it, but we're going to go back and get another piece that will have the coral lavore on it.
Perfect.
Yeah. So this is quiver number 2. Can you check Q2 there?
Q2.
Yeah, quiver 2. Yeah.
And then press submit. Not yet.
Perfect. That'll be great.
Oh
yeah, there's
PRIMNOID.
You said what?
Yeah, that's fine. Yeah. With aplicofferin.
Just pick it up. It's looking good.
So
Yeah, there you go.
Stephen asked where's the keyboard that use spacebar to
Yeah,
take
so-
shots? It's this one, but it's not. That's
Call.
nice. It's the DVRO. Which is what's it? Oh, it is.
You just
Yeah, that's right. And then you can hit submit right away
Okay.
because we already have a piece in the quiver. Yep, perfect. If you can fit the whole branch in there, I think
Trying to get
that would be
the
okay also.
branches right there.
Yeah.
Nice.
Excellent. So I'll just make a note that's in 2 pieces.
The same here as well?
No,
No.
yeah, you can write a note in there. You know, previous sample was two pieces, both in quiver 2. You can never take too many notes.
Okay.
Yeah,
Maybe that's okay.
yeah, that's fine. Awesome. So that's a good sample. 006 quiver 2.
So Steve,
Yes.
when you have a second.
Yeah.
Okay.
You know, if you see something interesting for yourself, if you know,
Okay.
just hit the frame
I'll
grab
just
button.
hit it.
Yeah.
It's so fun.
It is. Seeing a
That
lot
was a
of
great collection.
interesting stuff.
I know. You know, I don't know if you ever read like books by Dr. Seuss when you were little, like One Fish, Two Fish, Red Fish, Blue Fish. Any of these books? They're like classic American books, but I always think to myself, you know, could Dr. Seuss have imagined that these animals were actually real? And here they are. They're pretty otherworldly, but you know, they're right here on this planet. It's just really incredible to think about.
Okay. So we're continuing on. We
Excellent.
just passed through waypoint 1, and you know, we're gonna aim for the top of the ridge that,
That sea star beating up the road?
and, yeah, brittle star. Actually, it's a crinoid and a brittle star on some kind of stalk, but the creature that made the stalk is not there anymore, most likely. Could be a sponge stalk, could be a coral stalk, I'm not quite sure. But yeah, ophiuroid and a brittle star, so or ophiuroid and a crinoid. So the crinoid is the one on the right hand side. The Ophiuroid is the one on the left. And I'm guessing Ophiocanthid for the family of that brittle star on the left.
Yeah, Steve, that's what I think.
So yeah, pay attention. Boot
Nice
camp.
job.
Yeah. We had a pretty intense training the first few days of our transit out here on the ship while we've getting everyone trained up on coral sponge and associate identifications. Okay.
Did you
Onwards
see
and
that
upwards.
thing?
Yes.
It's just like a small black.
Yeah, I think what we're seeing, these small little discolorations, these nubs on the rock, and so you might see many of those. Very good.
I need a picture of you two. Look at me.
Oh, there's another one.
Perfect timing. Okay. Good.
All right. So, yeah, it looks like a small black coral down in the 6 o'clock position. Looks like bathypathes.
So black corals, they are dangerous to the reef or what? No, they just like being another type of coral.
Yeah, exactly. Yeah, a lot of the like reef forming corals, those are the stony corals. We haven't seen any of those yet on this dive, but you wouldn't really see a lot of reef forming species
Yeah.
down deep. They're maybe limited to 100 meters. Much shallower though.
There's a little thing right here.
Oh yeah, there you go. So sometimes when these crinoids are disturbed, they do this swimming dance. They
Dancing
did
over there.
they did this in
Twitter? No.
Why?
yeah, this is great. Oh wow, that's a massive sponge.
Yeah.
It's huge. That sponge is probably close to a meter tall, I'd say, if you measured it from the surface of the rock. Width-wise, it's probably 30 or 40 centimeters across the top of the osculum there. Very cool. Onwards. Oh yeah, there's a.
Yes.
Is there a chance we can look at that one?
begging for you
Lower
to
left
more
corner.
and louder.
Or we'll
Okay.
upper left corner here.
Get closer to the mic.
Okay. Sure.
We've got this might be a good candidate for an associate.
Is that another coral as well? Like right
Yeah,
there?
the one that we're aiming for is this pink, very highly branched colony right in the middle of the screen now.
Mm-hmm. Do you,
a Chrysogorgiid Octa Coral, so related to most of the corals we've seen so far. You know, these very
Chrysogorgia that looks different, it's young, and then like that, and then it starts getting bigger.
it does, yeah. There's a one called Metallogorgia you guys know about. When it's very young, it has these small branchlets. Zooming in.
Steve, you win. You didn't get skunked.
What did I win?
Well, you said we'd find small Chrysogorgiids here.
Yeah, this is not was
No.
most likely not Metallogorgia. This
But we
is a
weren't looking for-
Chrysogorgiid.
Is that a graph?
Oh yeah.
Right, but this is what I was.
I was not expecting to see this one though. This is I was expecting to see a different one.
What is this?
So this is most likely a species of Chrysogorgia, but I can't place it right now. Any
Look
finer
at
than
that.
that. I'm gonna go in a little bit closer for Tim since we've
What
got
kind of what kind of crab do we have on there?
yeah, that's a great shot there.
Hey Tim.
Since it seems to be very mobile, so maybe we can get the shot he wants of the underside since that's what's
Kind of
showing right now.
Paris Islet, I think, maybe?
So, for the folks on shore, we're looking at a Chrysogorgia, a golden coral is the common name for this coral that we're looking at, but also our group is very much interested in the associated organisms, namely the shrimps, the squat lobsters. Hey, there's an anemone on the 6 o'clock too, middle of the screen. So I've got two associations here. Let's see if I can focus on the anemone a little bit.
We might have to do that one.
That's all right. Yeah, it's moving around.
I think it is too.
Yeah, Steve, that looks like a Europe Tychus.
Europe
Paris
Tychus.
Talon.
Europe
That's a great shot right there.
So Tim, is this adequate for your needs?
Yeah, that looks great.
Okay.
I've actually never seen that kind of a branching pattern on a Chrysogorgiid, where it goes so far down the main
But I don't know
trunk
if we
of
want to
it.
take the first one or wait for another one. I hate it when I hate to.
Um, how big is? I mean, say.
Like
rough,
the
yeah.
whole thing?
I mean,
Probably looking at it's probably a meter.
yeah.
It's long. Can we just take the top half? I don't have to.
Not sure. I'm not sure we'll get the associates with that since that's about 2/3 of the way in.
Okay, I'm gonna. I'm here.
Awesome. Okay. So you can see actually. Look at the all the polyps closing. You can actually see they're closing very quickly, starting with the bottom half of the colony.
Do you want me to keep on taking?
Yeah. Some frame grabs of this. That way we can make sure that since it has multiple associates. I think this is very reminiscent of one, not the same, but reminiscent of one we collected in 2017. Yeah, I
Just don't remember.
vaguely, I can show you later. But still, yeah, these bushy, very highly branched, very interesting.
Yeah.
So we're gonna go into which biobox? I have A, B, and C here.
Well, it's farthest to the right.
Farthest.
It's got an H written on it.
Okay, thanks. So this is sample 007. And we're gonna go into the starboard biobox. So you can hit sample now. Sample store and then enter. Yep.
Nice job.
Awesome. Looks great. This will satisfy a bunch of our scientific goals for the dive. Yeah. Very nice. So
It got locked
box
up.
H. just write a note there. I forget what we